Hi, my name is Courtney Anderson, and I'm an application scientist here at ACD. Today I will be discussing a study that we conducted in collaboration with the Drug Safety and Development Group at Pfizer in Groton, Connecticut. And it's entitled The Fully Automated RNA in Situ Hybridization for Proliferation and Apoptosis-related Markers in Preclinical Animal Model Tissue Panels using RNAscope LS 2.5 assay. Preclinical drug safety assessment in animal models has been well established as a routine laboratory technique to evaluate pathological alterations that are induced by novel therapeutic agents. Traditional histopathological techniques such as hematoxylin and eosin staining or H&E stain, as well as immunohistochemistry or IHC, have been used to evaluate specific biomarkers and general morphology of the tissue. The IHC assay has been commonly used, however, it has several limitations, including consistent and systemic application of IHC with inconsistent performance due to various antibody clones, time-consuming antibody development and validation, as well as general lack of reagents for some animal models. RNA in situ hybridization technology, or ISH, presents an attractive alternative method for pathologic evaluation of biomarkers in tissues from preclinical animal models. And this is because nucleic acid probes can be specific for any biomarker associated with drug toxicity or a mechanism that can be developed and validated rapidly. Now, the RNAscope technology enables detection of almost any RNA biomarker with single molecule detection sensitivity and high specificity in formalin-fixed paraffin-embedded tissues. It provides a universal solution to characterize the tissue distribution of drug targets, biomarkers, in a highly specific and sensitive manner without the need to wait for antibody development or validation. And this assay allows visualization of each individual RNA molecule as a punctate spot under a standard bright field microscope that can be quantified. Now here we conducted a study to evaluate cell type proliferation and apoptosis biomarkers in a set of preclinical animal model tissues that are routinely used in toxicity studies. Now we used 25 different tissues, as we've highlighted here in this table, from 3 different species: dog, monkey, and rat. And we also analyzed several different probes in this study that I've highlighted here in this table as well, and we'll discuss in detail in a little bit, including some housekeeping genes, proliferation genes, cell surface markers, as well as apoptosis-related markers. Okay. Now I'd like to point out that we prepared our tissue according to the Pfizer-Groton protocol, which is a specific protocol in which they cut tissues into 3 millimeter sections and then fix these tissues for 24 hours to 48 hours in 10% neutral buffered formalin. So all the recommendations that we provide in the study here following this protocol. If a different protocol is followed, then optimization may have to be required for your tissue. We also performed this assay using the RNAscope 2.5 LS Brown reagent kit that is performed on a Leica BOND RX system. And we tested 2 different pretreatment conditions. One that's considered a standard pretreatment condition, which we performed for 15 minutes at 95 degrees using the epitope retrieval buffer. And we also performed a mild condition in which the epitope retrieval buffer is performed at 88 degrees for 15 minutes. And we compared how these 2 protocols worked for all of the tissues in our study. And I'd like to focus your attention now on figure 1. Here, where we show monkey testes tissue that was fixed according to our protocols I mentioned earlier. And we show the conditions using standard pretreatment as well as mild pretreatment, using our Polar 2A positive control probe and also our DAP-B negative control probe. And what you can see here is that while we have robust staining for Polar 2A with both of the conditions, the mild pretreatment condition is actually much better for the monkey testes. We still retain nice morphology and have robust signal. The standard pretreatment condition, we start to lose morphology. So for this type of tissue, we would recommend the mild pretreatment. In general, we recommend the standard pretreatment condition, but we have some exceptions. As I've shown here, the monkey testes is an exception where the mild pretreatment is preferred. And also listed in this table here, we have a listing of all the tissues that we recommend either for standard pretreatment or mild pretreatment. And this is for rat and most monkey tissues. We also have a couple of exceptions where for dog and monkey, the standard pretreatment would work well on these tissues listed here, and then the mild pretreatment would work for these tissues here. Okay. Now for the second part of this, we wanted to analyze several specific biomarkers and see what their expression pattern was like in a variety of tissues. For figure 2 here, we first looked at some apoptosis-related biomarkers in the rat testes. We looked at the genes FAS and BBC3, which are p53-regulated pro-apoptotic genes. And what you can see here is that we have very strong, similar detection of both of the markers in the rat testes. Our polar 2A also displays nice expression in the rat testes, and our DAPB negative control is clean with no background staining. For the next experiment, we looked at the cell cycle marker, Cyclin E1, in the rat tissues. We looked at lymph node and spleen, which were performed with mild pretreatment, and then we looked at the rat testes, which was performed with standard pretreatment. And we have very nice specific expression of Cyclin E1 in all 3 of these tissues. And then we also looked at some proliferation markers. One example of a proliferation marker is Ki67, which we show here in several different tissues from the 3 different species. We show the duodenum from the rat and the monkey and the dog. And we also show lymph node from rat, tonsil from monkey, and testes from dog. And in all 3 of these cases, we're detecting very strong, robust staining for Ki67. Okay? And then the last marker that we looked at was CD68, which is a marker on blood cells and monocytes. And we also looked at the expression of PCAM1 or CD31, which is an endothelial cell marker. And as you can see here, in the rat liver, colon of the monkey, and colon of the dog, we have detection of CD68, as well as in the heart of the rat, kidney of the monkey, and liver from the dog, we have specific detection of PCAM1 on the endothelial cells. So overall, these experiments demonstrate that we can get specific detection of several biomarkers in the various tissues from multiple species. And one last experiment that I'd like to point out is the quantifiability of our assay. So this is an original image here, and this is an image that's been analyzed by the Halo Imaging Analysis software system, where we can actually quantify the number of spots per cell in our tissues. This was an example using the Ki67 marker, where we can detect the percentage of total cells that have 4 to 10 spots per cell, and that equates to about 17.6% of the cells in this section. And then we also can detect cells that have greater than 10 dots per cell, which is about 12% of the total number of cells. Okay? So overall, what we've shown you today is that we have the ability to detect several different markers in several different species using either a standard or mild pretreatment protocol. So we provided you with an optimal protocol for using various tissues and also the analysis of several different biomarkers. And this demonstrates the utility of RNAscope for preclinical safety assessment studies.